But Michael Lynn is going to tell us about these dynamic filtering mechanisms in a inhibitory serotonin network and their nonlinear behavior. So Michael, the floor is yours.
Great. Yeah, thank you very much. Yeah, I'm excited to be here and present my my work. So here we go. So yeah, I'm going to be talking to you about an inhibitory serotonin network. And so this is work done during my PhD. Dr. Jean-Claude Bake is my supervisor, and this work was also done in close collaboration with Richard, also at University of Ottawa. So yeah. So as a lab, we're really interested in dorsal raphe nucleus. This is a really fascinating midbrain structure. It's involved in effective processing. And it's interesting because it's a network hub. So it both receives inputs from large portion of the brain, and it also sends outputs to serotonin outputs to a lot of the brain. And so these are just some of the structures it communicates with. And obviously, it's the target of antidepressants like SSRIs. So it's really important to understand how it processes input. So another structure I'll be talking to you about today is lateral habenula. So it's shown here. It's right below hippocampus. And habenula really represents negative valence signals with a really acute rate code. So it's really nice like that. So this is some monkey work showing that an air puff can really acutely increase firing rate, and these are graded responses. So depending on the strength of negative valence signal you give, there's really a graded response in habenula. This is just some more work showing that these neurons can burst very strongly. So we're really interested in dissecting this whole pathway and figuring out its involvement in mood processing and depression. And so just a brief overview, I'm going to talk about some synaptic physiology of this pathway. I'm going to be talking about dynamics of these recurrent serotonin connections, and I'm going to tell you all about that. I'm going to talk about information transfer through this pathway, and then finally some simple behavior and how we can use the predictions we've made with our network modeling to actually inform the behavioral results. So it's kind of a bird's eye view of things, yeah. So first, what does this input do? So we're just going to use some simple synaptic physiology first to characterize this. So we've injected habenula with an AAV expressing channel rhodopsin. We then make midbrain slices. We patch genetically identified serotonin neurons, and then we activate these habenula axons with short pulses of light. And so when we do this, we see something really classic. We just see a classic excitatory inward current. So this is AMPA mediated. It can be blocked with NBQX. We show they're monosynaptic. We also show there's NMDA components. So so far it seems like really classic, monosynaptic excitatory transmission from habenula to raphe. But there's actually something really interesting we started noticing, which is when we activate the same pathway at high frequencies, we see something completely different. So again, this is low frequencies. You have this excitation, but high frequencies. This is a separate neuron, actually. So what you see is at high frequency stimulation, you see an outward current. So this is an inhibitory current. And if you look at the time scale, it acts over almost a second. So really odd. We did some characterization. We did some IV curves. The long story short is we showed this inhibitory current is actually mediated by 5-HT1A receptors. So that's kind of odd. So this is just showing that an antagonist to 1A receptors, WEI-100-635, can completely block this current. So this is kind of odd because 5-HT1A receptors are classically thought to be autoreceptors. So that means that it's thought that serotonin neurons essentially regulate their own firing rate by secreting serotonin in the extracellular space. It binds to these autoreceptors, and it effectively provides gain modulation of their firing rate. So that's the possibility up top. We actually show there's a recurrent inhibitory structure happening in this network. So that's the bottom. So these neurons actually inhibit one another. So we have sort of a lot of convergent anatomical and other evidence for this, but I'm just going to show you sort of one piece of that story that's probably the strongest evidence, which is if you inject an opsin directly in raphe, and you inject it sparsely such that you have a sort of a salt and pepper mix of expressing serotonin neurons and non-expressing serotonin neurons, then if you record from one of the neurons that expresses this opsin and then activate the opsin, then you see nice spiking behavior shown here. But if you patch one of the non-expressing neurons, if the circuit is organized as a recurrent inhibitory circuit, then logically you should be able to patch a red neuron, a non-opsin expressing neuron, and still they should receive recurrent inhibition from the green cells. And that's effectively exactly what we see. So if you patch a red cell, you see this identical outward current, and you have no light-induced artifacts. So these neurons are completely non-expressing. So this is pretty strong evidence that there is actually a recurrent inhibitory circuit. We actually also completely rule out autoreceptor activation. So there's actually no component of the response that's due to autoreceptors. So next is kind of my favorite segment. So now we're going to be talking about information transfer through this circuit. So this is really cool. We found some really nice rules for this. So first of all, facilitation dynamics. So I just told you that high frequency activation produces this inhibitory response, but low frequency doesn't. So let's sort of dissect that a little more. So here we're still looking at raphe, but we're electrically stimulating serotonin axons while patching a serotonin neuron. We have on board a cocktail of drugs to block everything else. And here we show that we can elicit an outward current, still blocked with the same drug. So it's still the exact same thing we were studying before. And we can do these frequency experiments where we keep the pulse number fixed, so still 8 pulses always, but we vary the frequency. And what we see here is pretty striking. So at 1 hertz, you see absolutely no response at all in these neurons. And as you increase the frequency, all of a sudden you start getting this really massive inhibitory response. So it's a really steeply frequency-dependent response. Here's just some group data. This is kind of a pain to characterize, as synaptic physiologists will know. You can't take the amplitude, so you have to integrate over current to get charge, and then compare that to a surrogate noise distribution. But you can get some nice results out of that. So from this, we really want to figure out what the role of this is for network processing. So we made a model of these short-term plasticity dynamics. So this is actually taking a model developed by Julian Rosbroich, who was actually presenting at the same time slot in another session, unfortunately, and Richard No. And so it's a linear-nonlinear model of dynamics. So effectively, there's a presynaptic spike train. There's some kernel, which we show here on the right, which we've actually fit to the data. And then you pass that through a nonlinearity and convolve with a postsynaptic response, which we've also fit to this data I've shown you. And when you do this, you get something that's really fascinating. So at 1 hertz stimulation, this is just showing presynaptic spike train, in black. Then if you look at the bottom, the GERK response shows almost no outward current, effectively. Whereas at 20 hertz, you actually see this really large conductance start to appear. So this really maps nicely onto what I just showed you. I don't have time to go through all the data, unfortunately, but we also did some classic paired pulse experiments. We show that this STP model fits this data almost perfectly. This effectively could act as you could think of this as a training set, because this is actually the data that we fit the kernel to. We also tried to use this model to explain a completely different set of experiments where we found essentially meta facilitation. So really odd dynamics where the past history of facilitation influences the future facilitation possible. And so this data was not fit to the model at all, but we show that the model can almost perfectly explain it. So we have a really nice correspondence between the model and the experiments. So next we want to look at what I've described these really odd plasticity dynamics. What does this actually do for the network function? And so to do this, we made a sort of a toy model of raphe, just receiving habenula input. So these are 5,000 serotonin neurons. A subset of these receive habenula input. And most importantly, the recurrent inhibitory connections actually have the STP model I've just described in them. So the entire model is actually put into these. And we show that this short-term plasticity can actually support nonlinear input-output transformations, which is pretty interesting. So just to give you a sort of a taste of this, at 5 hertz, habenula input shown here, you really get a sustained serotonin neuron response and almost no GERK. Whereas at 20 hertz, you actually get an early peak followed by a really profound decrease in firing rate, effectively, which goes almost to baseline. And this is because the STP dynamics start kicking in, and you get an absolutely massive inhibitory response. So this is just characterized down here. And so what you see here effectively is the late response. So this 200 to 400 millisecond segment with STP, you really get a crash in the response as a function of the habenula output, right? And if you take out all the STP rules I just told you about, but keep the recurrent network in place, all of a sudden this becomes completely linear. So it really modulates this input-output transformation. Just to tell you briefly, another really interesting thing that these recurrent inhibitory connections with STP can generate is input gating rules. So what does this mean? If you have a network where part of it receives input from habenula and another part receives input from input A, you can then, so here on the left, you see input A can generate a nice response in serotonin neurons, but if it's preceded by input from habenula, shown on the right here, then you effectively get complete removal of the response of A, and that's shown here. And if you take out STP, all of that goes away effectively, and you have no modulation of separate parts of the network effectively. So from now, up till now, I've told you that at low input frequencies, you really have direct excitation that dominates, and at high input frequencies, you have recurrent inhibition. So what is the relevance really for the serotonin system would be graded threat processing. So I'm going to very quickly go through a simple head-fixed classical conditioning task. I don't have time, I think, to do it all, but we wanted to test the frequency predictions effectively for processing and behavior. So here, simple task, the animal's head fixed, it gets a cue, and it later reward, and it learns to anticipate. So here, just showing the licking, anticipatory licking, there's more for large rewards. And then what we can try to do is stimulate this pathway at different frequencies and look at the effect on modulation of anticipation. And so what we see here is that, if you stimulate this pathway at 20 hertz, you effectively get almost complete suppression of the licking response, of the anticipatory licking response. So that's shown in blue compared to black as non-stimulated trials. And at 5 hertz, you actually get absolutely nothing. So this maps really nicely onto the short-term dynamics I've just told you about, right? Where 5 hertz excitation would dominate, 20 hertz inhibition would dominate. This is just some group data here. And so the conclusion really is that there's this recurrent inhibitory network acting over behavioral timescales and really governed by these short-term dynamics. And it filters aversive information to modulate some sort of behavioral anticipation. And so I just want to say thank you for everyone involved, especially my supervisor, Dr. Jean-Claude Bake. Here's other people involved. You can follow me on Twitter if you want, or ask any questions. So yeah, thank you.
Thank you, Michael. That was a really lovely demonstration of modeling and physiology. It was a beautiful combination. And
yeah,
we have people who are just really shy in the audience, but it's not going to stop me from asking or commenting.
Okay.
I think that this is your work shows how important it is to be testing across all the range of frequencies. And I wonder to what extent how much of this kind of behavior has been missed. But it occurred to me, I mean, you're looking in Raphe and habenula networks, but it occurs to me that this is almost like a cellular protective mechanism. For instance, in epileptogenic tissue, you have a runaway effect where there is no inhibition. And it just occurred to me that maybe something like this might be acting elsewhere to protect people from these kind of epileptic-like phenomena. Now, what do you think about that?
I mean, yeah, that's certainly really interesting. And I guess it is true that raphe itself has a really low firing rate of a few hertz typically. So that's why it's thought that this current was actually auto-inhibition. So the cells inhibiting one another. I think it could still play the same role you're describing at the level of the network, right? Even if the neurons aren't directly inhibiting themselves, they might inhibit their neighbors to sort of regulate overall excitability. So yeah, that's a really good point.
Yeah, you might want to try testing it with anticonvulsants,
Yeah,
just for interest.
yeah.
Okay. Well, thank you so much, Michael. That was really great. And
you.
so now we move to our last speaker.